Welcome to, I guess this is disease hide and seek. And that's actually a really good name for this topic because we're going to be talking about risk assessment, disease risk assessment in crops. And, you know, probably since the very first hunter-gatherers started producing crops, they tried to predict the risks of things like diseases and pests. Today, we're really good at telling you what causes it. We can tell you disease and life cycles of the organisms that cause the problems. We can even show you pictures of ultrastructure of the interface between the pathogen and the host, or the molecular determinants of virulence and pathogenicity. But I still can't always answer the question, should I spray my canola for stem rot or not? So that's really what we're here to talk about is, you know, we know so much about diseases. How come we can't accurately predict them every year? How come one year this disease is so bad and the next year it's not, and we can't always give you a great explanation for why that is? From an applied standpoint, really what we want to do is get better at measuring or assessing the risk of a disease. For a couple of reasons. The first is, when my kid gets sick and I take him to the doctor, they assess the symptoms, they look for signs of things, and then they treat the disease and they cure it. Is that what we do in crop disease? Do we wait for symptoms to show up and then intervene and cure the disease? No, we don't do that. Once the symptoms show up, we can't cure those. So that green tissue is lost to the disease. So really, in crop diseases, we don't take a wait and see approach. We essentially apply all of our management tools for most diseases before we even see the symptoms of them. So if we have a good diverse crop rotation, if we're deploying genetic resistance, if we're using good agronomy and other things to help have a healthy crop, all of that stuff we do before the disease is even initiated. So some of our very best disease management tools, we deploy them long before we ever see the symptoms. Now there are some in-season things we can do, like apply fungicides, but similarly, because our fungicides are preventative or protective, we have to spray them before the symptoms show up because we can't cure diseased tissue. We can only protect green tissue. So as a result of that, we have to just take our best guess as to whether there's a disease risk. So why is it so hard for a plant pathologist to tell you if there's going to be a sclerotinia risk in your canola crop? Well, there's a few reasons for that. One is, what I can tell you is that disease happens when all three sides of the disease triangle come together. You got a susceptible host, a virulent pathogen, and an environment that's conducive. So we don't really have true genetic resistance for sclerotinia. Pretty much all of our crops are susceptible to it, our broadleaf crops. We know the pathogen is endemic to Alberta, but what we can't tell you about is what the weather's going to do 3 weeks from now. So because of the challenge in predicting the weather, we can't always know what these diseases are going to do. The second thing that makes sclerotinia so challenging to predict is that you almost have to kind of think of two separate disease triangles. One for the germination of the fruiting structure that produces the spores. So are the conditions right for apothecia formation? And then later on, are the conditions right for those spores to land on the petals at the right crop stage, and is there enough moisture for them to initiate disease on the crop? So when those two things come together at the right crop stage, and the weather's just right, then you're going to get disease. So that's challenging to predict. Okay, how can we get better at evaluating risk in crops? Well, there's a lot of new technologies that are being brought to bear to get better at this. And I've got some posters here that we're going to just briefly go through, and then you can spend time reading them or take pictures of them. I've got information on the table about crop diseases. Feel free to graze that. Or we can just have a question and answer and a discussion about some things. So that's where we're headed. So the first thing I want to show you is a project that I'm not involved in, but it is This poster was prepared for Canola Palooza, which takes place next week in Lacombe, which is a great meeting that you should consider attending. This is a group of Ag Canada scientists that are using satellites to predict sclerotinia risk. So it's quite interesting. They, from space, can collect information on when the crop is flowering, so they know when the right crop stage is for disease risk. They can detect soil surface moisture and the days that the soil stays wet. They can tell you the last time a susceptible crop, canola crop, was in that field. And by putting together this information on temperature, soil moisture, precipitation, years since the last canola crop, and some user inputted information, they use this disease risk tool, the DIRT. I thought that was funny. I guess you guys didn't. But anyway, they collect all of this information and they do their best job to predict that there's a risk that sclerotinia, all of the conditions are right. You are there for a high risk. So for example, there was a susceptible crop in that field last year. There's been a lot of precipitation. The soil has stayed wet for a long time. The canola crop is coming into flowering. All the alarm bells are going off now, so you got to beware. Okay, so I'm going to leave that one now. Feel free to have a closer look at this a bit later. This is a project that I have been involved with a little bit. This work was done mainly by Ralph Wright's group. He's an ag meteorologist with Alberta Agriculture, and Brian Kennedy from the Wheat Commission was instrumental in moving this forward. So some diseases are, like sclerotinia, are really, really weather dependent. We know the fungus is here. We know we don't have true resistance. So it's really up to the weather to decide what's going to happen. And so a disease that's similar to that is Fusarium head blight in wheat. The Canadian Grain Commission reported that between 30 and 40% of wheat samples in Alberta were downgraded due to Fusarium damaged kernels in 2016. So 30%. And then the next year in 2017, it was less than 1%. And so the difference between our summer of 2016 and summer of 2017 took us from 30% of samples downgraded to less than 1%. So it's incredibly weather driven. And so we can use weather data to sort of predict the risk of Fusarium head blight. And so Alberta happens to have one of the best weather data collection systems in the world, thanks to Ralph and his group. And so it was very easy for them, once they got the model, to just use all of that weather data to create maps that show you where high risk and low risk is. So if you Google ACIS, Alberta Climate Information System, you'll get their page, and then on the right-hand side, there's a tab that says mobile users or something. Or there's a tab on the left, I think, that says Fusarium risk, and under the maps section. So you can pull up a map that gives you a dashboard-like display for where the risk is high. And as the risk increases, the map turns from yellow to orange to red. You can also pull up, so this is an individual dashboard for one weather station. So you can choose a weather station to look at, although the tool will automatically go to the weather station that's closest to where you are if you allow it to use your location. You can look at the Fusarium risk in the last few days. You can look at precipitation and temperature and all kinds of things. But basically, when your crop is getting ready to come into heading, you'd want to be looking at this tool and seeing if the risk is high. Keep in mind it's not a forecast. It's just the actual weather data that's collected. It's not forecast, not a forecasting tool. But if there's been a high risk of Fusarium and you're coming into heading, you want to be ready to deploy a fungicide, depending on the risk of Fusarium in your area. Here in southern Alberta, this could potentially be a real important tool. Now, one of the questions that was asked was, how does irrigation affect the use of this tool? And the answer is we don't know yet. This is the first year that this has really kind of been deployed, and there's still some ground truthing to do and things like looking at the effect of irrigation So unfortunately, we don't know that yet. Okay, moving on to the next one. I mentioned that for diseases like Fusarium and Sclerotinia, we know that our crops are susceptible because we don't have strong resistance in them. We know that the pathogen is there. We can monitor the weather, but sometimes the fungus isn't producing spores at the crop stage that gets infected. So during the bloom period, if Sclerotinia isn't producing ascospores, then there's little risk of the disease getting started. So unlike insects, where we can kind of sweep the crop and then look in the net and identify what's there, we can't see these little microscopic propagules with our unaided eye. But there are some technologies that can do that for us. And so that's what the last two posters are about. This one is on biosensors. And Susie Lee at Inotech in Vegreville is working right now on a biosensor for Sclerotinia. And essentially this is a little instrument, you know, about this size that you would attach to a stake and then put in your field. And then as the Sclerotinia ascospores just passively float and impinge on this detector, or I should say receptor, there's a combination of biochemistry and engineering and pathology that goes into transduction of that into a signal that comes to your phone. So once a threshold of spores that's dangerous for disease initiation is reached, it will send you a message to your phone saying, uh-oh, I put here time to spray your canola field at that location. That's not what it's telling you. The spray decision is up to you, but this is information you can use to make a decision. I now know that my crop is coming into flowering. My biosensor just went off. I know I'm at a high risk, so that helps inform me on what I'm going to do as far as a spray decision. Okay, last one, spore trapping. So this is a project I've been closely involved with. And so my lab in Brooks, in partnership with Promax Agronomy out of Lethbridge, has been looking at a way to help potato growers know when there's a risk of Phytophthora infestans, which causes late blight, which is an extremely aggressive and devastating disease in potatoes. And so my partner here, Lori, is going to talk about spore traps and how they work, and she's going to tell you a little bit about the spore trapping network that has been set up for potatoes. And this, so right now, this trapping network that we have is we're just looking at potato diseases. And the reason for that is because the Potato Growers of Alberta is paying for this. But this type of technology could be developed for other diseases on other crops. And in fact, that is happening. So Andre LaRoche at Lethbridge is looking at doing this for stripe rust. Shama Chatterton is looking at doing this for Sclerotinia and dry beans. Kelly Turkington is looking at doing this for wheat diseases at Lacombe. So it's kind of an up-and-coming thing. And what would be really great is at some point in the future, if we just had a network of spore traps across the province, and like this weather tool, you could just go online and say, where's the nearest spore trap to me and what are they finding in it? That's, hopefully we can get there at some point. We're not there yet, but Lori's going to tell you a little bit about how this trap works and how our network has functioned.
Okay, so this is the spore trap we use. We have 13 of them set up across the province, and we also have 2 additional traps that we can switch out in case there's breakdowns. So this fin part of it basically just helps make sure that it's getting the right angle at the wind. And in here is where it samples. So let's take this off. So it basically runs like a vacuum. So it pulls the air in and then goes into the vial. There's 8 vials in here. Each one samples for 1 day. So we have it set up to sample 3 hours each day. And so theoretically you could leave it sit up to 8 days and it would still sample continuously for those 8 days. But in order to have the data that the growers are getting to be timely, we send in the vials to Mike's lab twice a week. So we have, for the 13 traps, 8 different operators. Each person has 1 or 2 traps to look after. And we have the day staggered that we're mailing also, so that the lab doesn't have every single person sending on the same day. So basically all the air that's collected, everything that's in it goes into the vials, and then they analyze that for the spores. We discovered a couple years ago that if these are sitting near particularly seed canola fields, that leafcutter bees really love to crawl inside here, and then they hang out in the vials, which doesn't really make it easy to see the spores in the sample, and it's not very fun collecting when there are live bees in the vials. So there's the solar panel and then a battery inside along with a control panel inside, and so it stays charged all summer, theoretically anyway. Sometimes there's some issues, but it's very self-sustaining as long as you keep checking it to make sure it's running and collect the vials from it. From there, I send out to potato growers a report once a week, so this is a little part of the report. So we do a summary of late blight across the province, and I break it down also into each particular trap, talk more specifically about what was found each day for each trap, and there's also a live document that the data gets put into, so growers also have access to that document so they can see more than just the once a week what is happening in the trap that is nearest to them. Did I miss anything?
No, I think you got it.
Okay.
Okay, any questions?
What's the approximate cost of one of those?
Last time we ordered, I think it worked out to about $8,000 each.
So when the samples get sent into your lab, Mike, you can differentiate what types of spores they collected in.
So some of the things flying around in the air that are going to cause disease are readily we can recognize them very readily under the microscope. And so Phytophthora infestans, well, I should clarify that we can't distinguish between different species of Phytophthora, but we can tell if it's a Phytophthora sporangium. So that's what this is right here. There's a couple of Phytophthora sporangia that under the microscope. So if the pathogen has a diagnostic propagule that we can recognize under the microscope, then we can report it. And so we actually on the report, in addition to late blight, will also tell you how many alternaria spores are flying around. So alternaria causes a couple of diseases on potato. And we can also recognize Fusarium spores under the macroconidia under the microscope. So there are a few different things that we can recognize. Now, for some pathogens, like Sclerotinia sclerotiorum, I can't tell one of those ascospores from. There's a bunch of things that are just little round, clear balls, or sometimes they're a little bit oblong, and they're not very diagnostic to look at. So for things like, so I guess before I go there, I'll say we are working with the Alberta Plant Health Lab in Edmonton to take this from a microscopic evaluation to a quantitative PCR evaluation, where instead of looking at each sample one by one under the microscope, we just, if 15 vials come in, we just use the contents to prepare a quantitative PCR reaction and run all 15 at the same time to get an estimate of the numbers in that sample. So that could be done for things like Sclerotinia, where we can't really easily recognize them under the microscope, but we could use molecular tools to sort of quantify or semi-quantify the amount in the air. So again, hopefully at some point in the future, these samples are coming into a lab. The lab has a barrage of tests that they run on the sample, and then they can tell you how much Fusarium graminearum is flying around, how much Sclerotinia sclerotiorum is flying around, how much, pick your favorite disease, is flying around. And again, this just gives us a little bit better idea of what the risk of that disease is in a certain area. Now, the reason this has been so successful for late blight is that Phytophthora infestans, when it produces sporangia, they very quickly get up into the air and they can blow for miles. They're very adapted to aerial dispersal. Sclerotinia is not the same. It doesn't get up in the air and float for miles. It floats for maybe tens to hundreds of meters. So it's more of a localized dispersal, and that's going to affect the number of traps needed, the density of traps needed to really be predictive. So Shama Chatterton's going to look at sorting some of that out in the project she has on dry bean. So there's still a lot of these, pretty much all of these I'm talking about are very early stage tools, but hopefully they will develop into something like you could purchase a biosensor and deploy it in your field, and then it'll notify you. Or sometime in the future, a trap network that you could go online and see what's been detected in the trap closest to you.
Right, have we been looking at the possibility of a biosensor for leaf blight?
Yes. So it's too bad Russ isn't here, because he actually was in contact with a company or a researcher out of the UK that reportedly has developed one. And we were hoping to deploy some biosensors for Phytophthora infestans in southern Alberta this year, but I don't think we've been able to get our hands on some. So if that technology doesn't exist, I think it's very close. Yeah.
How close is Susie to releasing a biosensor?
I wish I knew. I'm not sure. This is going to be a very easy, it'll look like a really simple little instrument, easy to deploy, but the biochemistry and the engineering that go into producing this is really complicated stuff. So when I sent this to Susie and included her as an author, she sent me back, "I like your picture." She was being kind, because this really oversimplifies the work that she's doing. So it is a very complicated procedure to, I believe hers is an immunological based test, so antibodies, specific antibodies that will recognize spores. And I believe that what happens is that when the spore binds to the antibody, it changes the electric field across the receptor. And once enough spores have been bound to the things, it changes the electric field enough that there's a signal. And then that signaling goes to your phone. But Susie could tell you all kinds of amazing details about how that works. It's really complicated. I haven't heard an estimated release date. Okay, if there's no other questions, feel free to have a look around. There's lots of information on other crop diseases. And the last thing I'll show you is that we inoculated some of the plots back here, but because their crops are so young, there really isn't a lot of symptoms to see. But we do have some potted wheat here that has Fusarium graminearum symptoms at the crown. So lots of times when we talk about Fusarium graminearum, we're talking about head blight and the symptoms on the head. It's important to not forget that this pathogen actually is really good at colonizing this part of the plant. And so you can see some symptoms here. So even in a year like 2017, when we didn't have a lot of head blight, the fungus is still there. It's just down low and didn't get a chance to cause head blight because there wasn't enough moisture to initiate a head infection. So have a look. You can feel free to pull these apart and look at them to your heart's content.
Does the root blight infect the head?
So does the root blight affect the head? Depending on how early the infection happens and how extensive it is, if the infection kind of girdles and chokes off the plant, then there will definitely be major effects that you'll basically fail to get the plant could die, or at least you'd fail to get a head with anything in it. If the infection happens later and it's not really severe, it could, then you get the whole spectrum, all the way from a dead plant to a very minor effect on yield. But yeah, depending on the environment, when the infection occurs, what kind of resistance or tolerance the plant has to it. But yeah, it can be anything from a dead seedling to a plant that has just kind of minor effects.
It doesn't cause Fusarium head blight.
Okay, so excellent question. So the Fusarium graminearum that's colonizing that bottom part gets splashed up or blown up onto the head. And if it does that during anthesis or during a certain part of the heading stage of the crop and there's moisture, then it will cause infection and cause head blight. So it's the same thing. It can cause root rot, crown rot, seedling blight, and if the conditions are right during heading, it'll cause head blight. And Fusarium graminearum is the species of Fusarium that is best adapted to cause head. It's the most aggressive head pathogen. So Fusarium avinaceum, Fusarium culmorum, Fusarium equisete, all of these other Fusarium species can colonize the root and the crown. Sometimes they get up onto the head, but they're not nearly as aggressive, and they don't cause nearly the same amount of head blight as Graminearum does. That's the force to be reckoned with as far as head blight.
So fungicide application, that heading isn't going to do anything for your root blight then?
No.
You're just protecting that.
Yeah. So how would we do our best to prevent the significant root or crown rot? Yeah, all those good practices that we deploy before the season even starts. Select a cultivar that has some tolerance, use a good diverse crop rotation, good agronomy so you have a healthy crop, and then in season, if there's a risk of head blight, then we apply a fungicide. Yeah.
Could you have an early application of fungicide? Would that protect the root blight or no?
Yeah, so there's been a lot of work done to see if applying fungicides with herbicide, like at that herbicide timing or early, how much effect that has. And yes, you can knock disease pressure back a bit, but generally speaking, there's little yield advantage. So we can't really protect a lot of yield. And one of the reasons is that if there's going to be damage done, it's probably going to happen at the seedling stage or even at germination and emergence. And so with the exception of seed treatment, there's probably not much we can do. Hopefully people are using good quality seed that doesn't have a lot of disease on it, good vigor, it's going to come out of the ground and get a good start. And then in those cases, we don't see a lot of advantage to spraying fungicides early. When we do see an advantage is when we protect the flag leaf. So spraying at flag leaf emergence up to the end of heading time is really the window for applying fungicides that are going to protect yield. Did I miss any, Ron, did I miss any management tools for preventing root rot and crown rot in cereals?
Using disease-free seed and seed treatments, obviously the most important protecting the seedling health. Crop rotations you've mentioned, there are certainly some differences in tolerance to head blight amongst the varieties of wheat and barley and Ag Canada and Lacombe. Telly Turkington puts out a nice chart every year so we actually have some varieties now that have a tolerance to Fusarium head blight, which was a tool we didn't have years ago. Yeah. And then of course your fungicide management. And another thing that's very pertinent to us here is irrigation management. If you irrigate at heading, you could be in trouble because you create that microclimate that Mike referred to that allows the infection of the heads to occur. So yeah, there's and then with Ralph Wright and his Fusarium risk assessment program, that's a brand new tool that we have this year. Because this is so much of a weather-driven disease, as Mike mentioned, understanding the weather and the role it plays is important. And I think Kelly Turkington, you can correct me if I'm wrong on this, Mike, was interested in the effects of the fungicides that are applied for the leaf diseases in wheat, as to whether flag leaf applications of those have an effect on head blight, because there's usually we're out there applying fungicides for the leaf disease as well as head blight, but some people are saying that they may be seeing a benefit on head blight with the later fungicide applications for leaf spots. Do you know any further?
Yeah, so Kelly and Randy, and we had a site at Brooks that was part of this study. We looked at if you apply fungicide at flag leaf emergence, or if you apply at head emergence, or both, what is the best case scenario? So when you applied at flag leaf emergence, and there was disease pressure, you got a certain amount of protection and a yield benefit. When you applied at heading, you got the same effect, and when you applied both at both headings or at both timings, you got significantly better suppression of disease than just one, but you didn't get more yield. And so the conclusion of that study is that on the prairies where we tend to have, especially in areas where the conditions are quite dry, we don't have high humidity or a lot of precipitation, fungicide application at heading is just as good as an application at flag leaf emergence. You get the same amount of yield protection. Now the only exception would be in years where, like 2016, where there's a lot of precipitation and disease potential becomes higher. The one caveat I'd say to that is if you're spraying fungicides at heading, you need to make sure that you don't use a strobilurin, because the strobilurin can actually induce the Fusarium fungus to produce more mycotoxin. So if you're applying fungicides at heading, make sure you're using the fungicides that are registered for control or suppression of head blight. And then you'll also get the benefit on the leaves, the foliar diseases. But we don't want to be applying strobilurins at heading because we can sometimes increase the mycotoxin concentration in the grain.
Could you talk about, Mike, about the latest on disease risk assessments and so on for stripe rust, because that's going to be the first disease that we're going to be contending with probably.
Yeah, so there's a few ways that we get a bit of an indication on stripe rust. Especially in cases where we're growing resistant varieties, we can take a bit of a wait and see approach. And so when the symptoms show up, then we know it's around and we need to protect it. But for people growing susceptible varieties that are at a high risk, you can't afford to take a wait and see. So Andre LaRoche is working on a spore trapping detection system similar to this for stripe rust. And the second thing that has been done in the past is that Kelly Turkington and Kei Kwan Shi at Lacombe are involved in a wind trajectory group. And so what they do is track parcels of air. And so when stripe rust, if it doesn't overwinter here, it usually blows in from the Pacific Northwest. So they'll track air trajectories and then they'll send out alerts letting us know that a parcel of air from the Pacific Northwest could potentially have deposited some spores, and that gives us about a week to 2 weeks advanced warning that stripe rust could be getting going. So right now those are the two that I'm aware of.
Has the stripe rust overwintered this year?
So Dr. Rima Bukador at Lethbridge has been out surveying winter wheat fields and hasn't found stripe rust has overwintered. We haven't seen any evidence of stripe rust yet. Has anybody seen stripe rust?
Sometimes
we
it
haven't.
overwinters better in central Alberta with
Yes.
the snow cover.
Yeah, that's true.
Have they been scouting up there as well?
Yeah, so I haven't heard, Kei Kwan and Kelly have not reported any stripe rust up there either. So it looks like we kind of finally got a winter this last season, and it looks like it may have kind of taken care of it for us.
So Washington State usually has a very active program in stripe rust monitoring, and I think perhaps yourself, Kelly, others, look at what Washington is reporting. What's the stripe rust situation in Washington?
I don't know. I haven't kept up with what's been going on there, but we know that it didn't overwinter here. We haven't had any reports of it in commercial fields yet, and we haven't really had any air trajectory high-risk situations. So we're pretty sure it'll show up eventually. Without a spore trapping system, we can always get surprised, but it looks like we haven't seen any evidence of early infections yet this year.
Mike, has the change from black tillage summer fallow to zero till change the patterns and the gravity of diseases?
Yes. So when we leave a lot of the crop residue, that's for especially diseases like Fusarium, that's where it overwinters. And so yes, But really the only high-risk situations would be when there's short rotation. So if you're not leaving enough time for the crop residue to break down before you bring in another susceptible crop, then there is an elevated level of risk. But you know, if you grow a wheat crop and you have Fusarium in it, and you don't bring you know, a barley or corn or wheat crop in for 3 years, that residue is pretty much all gone by then. So the risk that you left behind is pretty much eliminated by the time you bring the next susceptible crop in. So it can be a real issue when you're growing canola wheat, but if you have a nice, diverse, longer rotation, it's not as much of a problem.
Are the diseases also controlled by the breakdown of the organic matter?
Yeah. So for a lot of diseases, once that crop residue is gone, the disease risk really drops off. It can still survive in the soil, but there's not large amounts of inoculum that are going to immediately cause issues in the coming crop.
Are you finding any negative effects by using fungicides on microorganisms in the soil?
That's a great question. There hasn't really been a lot of studies there, but in light of how much more we know about the positive effects of beneficial insects, and how if we can hold off using insecticides, those beneficials do a lot of the work for us. There's been a lot of questions over the last few years about, well, what about fungicides? Are we making the situation a little worse every time we kill beneficial fungi with a fungicide application? And the answer is we really don't know, certainly not in southern Alberta, what the impact of, say, increased fungicide use would be on some of those beneficials. Presumably, anytime we add anything to the soil, it's going to change. I mean, if you pour water on the ground, it's going to change what the microflora, certain organisms are going to be better adapted to live in wet soils than dry and vice versa. So certainly when fungicide comes in contact with some of these fungi in a field, it could affect the balance of beneficials versus pathogens, but we don't really have any good assessments or good answers. There's been a couple of studies that have just started to come out, and I'm not familiar with the results of those studies, but it's a really good question, and it's one that comes up more and more frequently. And I think especially because of the dramatic difference, the dramatic effect of having beneficial insects. If we don't kill those beneficials with respect to insect pests, we can manage a lot of insects by just letting the beneficials and the natural predators do their work. So it's a good question. I don't have a great answer. We just really don't have enough information yet. Okay, so Ron mentioned cereal disease susceptibility sheets that Kelly Turkington produced. I've got them on the table here along with a bunch of other information. So feel free to look at the posters, ask some more questions, pick up any information you want. But that's really all we have to say. So thanks very much for your visit.
Thank you.